Okay, welcome everyone to today's Beyond the Scope talk. For today's talk, we're going to have Dan Huber talk a bit about FIB. Dan's one of the assistant directors here at CMAS and in charge of the FIB instruments. So he's always quite busy with all that kind of stuff going on. So he'll have quite a bit to say about the FIB. So if you're interested in that, definitely ask lots of questions either during or after the seminar. Generally try and keep these a little shorter anyway. So hopefully have lots of times for that. And then also if you have questions about building instrumentation stuff, I know Dan loves to talk about that as well. It's one of kind of his hobbies almost. So I'll just mention the other upcoming talks. So we're doing this every 2 weeks. In 2 weeks, Rob Williams will be giving a talk on some high resolution work in the TMs, and then move to cryo EM with Giovanna. I believe after that, we're going to go to the SEMs again with Steve. So I'll pass this on over to Dan now.
Well, thanks for the introduction, Dan. We want to talk about some serial sectioning and imaging in the FIB today. It's one of my favorite things to talk about. And I do spend a lot of time on the FIB, and I think doing TEM sample prep and serial sectioning is a lot of fun. I say serial sectioning is probably more fun than a lot of other things because you're finding something underneath the surface, and you're uncovering something or discovering something new. And when you start off the experiment, you don't necessarily know how it's going to turn out until all the data is processed and stuff in the end. So hopefully we'll have enough time at the end of the talk. I'd like to be able to answer people's questions and kind of customize what we've got in terms of capabilities and the knowledge for the folks that are here. So I appreciate everybody showing up this morning for the talk. We're going to be, as Dan was saying, we'll have various different topics in the discussion series here, but today we'll be looking at just the focused ion beam system. CMS has got a wide variety of instruments, from scanning electron microscopes, transmission electron microscopes, and the systems that I'm involved with primarily are the two dual beam FIBs. I use all the different instruments, but I'm the instrument manager for both dual beams. I grew up in Dayton, Ohio. I was a material science undergrad here at OSU for a while back in the early days, and finished my PhD in material science. I've got a material science background. Still doing a lot of materials characterization, and I work on a wide variety of materials, from soft materials and biomaterials, and still enjoy working on hard materials and titanium alloys and stuff. What am I really excited about in terms of the microscope facility? I've always spent a lot of time characterizing the electromagnetic fields, vibrations, temperature variations, and stuff to make two things: the dual beam FIB sample prep for the highest resolution, atomic resolution STEM imaging, but then also optimizing the environment so that the microscopes can perform and we can get the best image. So doing the atomic resolution imaging is something I just find really fascinating, and I try to work on incorporating all my skill sets to try and make the environment and the sample as best as possible for that sort of work. Got animation here. So what is a dual beam FIB? Dual beam FIB is our tool of choice, and why do we want to FIB everything? The dual beam FIB is it's based on a scanning electron microscope that also has a focused ion beam microscope in it. Now, each of those tools are available as individual instruments, and the dual beam instrument that we're really excited about has both the electron beam and the ion beam, hopefully focused at the exact same point in space, so that we can mill samples with the ion beam, which is a destructive process, but then also use the electron beam, which is a non-destructive process, to take a look at what we've milled and the material that we're removing. So scanning the electron microscope, your incident beam is going to come down, it's going to interact with the sample, and it's going to generate a number of different signals. Basically, photons can come out. We can have cathodoluminescence or X-rays, but we can also have backscattered electrons, secondary electrons, and Auger electrons, which are going to come from different parts of the sample and give us different types of information from those from the sample surface. The focus, the ion beam, if we want to like take a look at the difference between the electron and the gallium atom, is imparting the sample. An electron, if we said the electron was going to have about the mass of like a fly or a bumblebee, the mass of the gallium atom imparting onto the sample surface is going to have a mass to be equivalent of a locomotive engine. So you can just imagine with you're scanning the surface of your sample with these like little tiny electrons. They really don't do a whole lot to change or modify the surface. But the gallium atom, when it comes in, it's just got a tremendous amount of energy. It imparts that energy on the surface, and it can actually ablate and remove material. And so that's where it's kind of fun. You can kind of blast away material and do, modify your sample, dig into it, look underneath it, and change it at the nanometer or micron scale. So why do we use gallium for an ion source surfaces? Oh, you got the gallium-based FIB, gallium-based FIB. Other materials have been tried, but and used as an ion source, but gallium has a lot of benefits. The gallium is a liquid at very low temperatures, basically like near room temperature, and so that liquid is going to melt and then wet the tip of the needle that forms the ion source in the presence of a large electric field that produces what's called a Taylor cone, which gives us a very sharp source of ions. So it gives it a very high brightness. So we have basically a lot of ion flux coming from a very small area. So it would be analogous to having a very sharp knife. So it's a very good thing. Second point, gallium has a low melting point, but it also has a very low vapor pressure. And so that's going to allow us to have liquid metal in a vacuum. And basically that metal is not evaporating and filling the chamber. So if you had, say, for instance, you could use mercury. Mercury's not very good for you and everything else, but mercury has a very high vapor pressure. So if we were to put that into the microscope, pull a vacuum on it, the mercury would actually be evaporating and filling the chamber full of mercury vapor, which would be not desirable. Gallium has a very low vapor pressure. It gives us very high resolution in the vacuum. The lower the vacuum, the larger and the longer the mean free path of the electrons and the ions, basically better our resolution is going to be. Gallium, the sources that we use are actually a single isotope. Gallium has naturally forms and comes in two different isotopes, gallium-69 and gallium-71, and they naturally occur in a ratio of about 60-40. So these sources have actually been gone through the trouble of isolating a single gallium isotope. That means if you had two different isotopes going down the column and focusing, the two different masses would actually focus at slightly different positions. So if you had a multi-isotope source, your image would be just slightly more blurry than if you had a single isotope. Another benefit to gallium, it's relatively rare. It's not found in most minerals and materials. So if your sample does get some gallium in it from the process, generally when you're doing EDS and characterization and analysis of that sample, you can ignore the gallium signal because you know it did not come from your material. It's also a heavy ion and it produces a nice effective sputter yield. There are other atoms you could use that might sputter the surface. You put a lot of ion flux in, but it wouldn't yield very much milling. Gallium is effective in its sputter yield, so that's another advantage. Other sources, there are xenon and other plasma sources for FIB. They don't have the benefit of having that nice sharp Taylor cone to produce a nice bright source at low beam currents. Where they do excel is if you get up to very, very high beam currents in excess of tens of nanoamps, you can get a very high current with a slightly better brightness than the gallium source. So there are xenon and plasma source FIBs that are available, and one of the latest developments is now Thermo Fisher is producing a system that's got a xenon, neon, oxygen, and argon species. And it's actually switchable. So you can actually start the machine up and you'd be running in xenon, and you can switch the other ion sources for milling. That has the benefit that there's slightly different contrast mechanisms and sputter yields and stuff that can be exploited by changing your ion species in certain situations. This is still, kind of a new and evolving field, and the papers are just now starting to come out as people are exploring this. That's something we're actively pursuing, is being able to add to CMS in the future, but we don't have it yet. So stay tuned. We'll see if we're successful in our proposals. So to say, how big is a FIB sample, and how can I use the FIB to solve a problem? And the first thing people say is like, well, or we say, how big is a FIB sample? We say like, well, how big is a human hair? If this was a human hair, and how big would it be? I actually have an example where we cut into human hair to actually study some phenomena that was not well understood, and that was the formation of these little bubbles. There's basically calcium fatty acid that was forming underneath the layer of the hair. And if you think about a hair, the structure of it is a little bit like a tree trunk, where you've got the cortex and the cortical materials. It's kind of like the wood inside of a tree, and then you get this outer structure. It's the cuticle. It's a little bit like the tree bark. And so the problem was there's these little bubbles that were forming kind of under the tree trunk, or between the tree trunk and the bark. This had been seen before by just taking a razor blade and kind of slicing into material. And so it was kind of a good idea of where the material was existing and how it was manifesting itself as the bubble, but it had not been really seen in 3D. So the one benefit here, we have a 3D structure that we want to characterize, but just by looking at the surface, we've got kind of an idea already how big it is, where it's located, and kind of we've already got an idea. Our brain is looking at this little bubble and getting a picture of what we think is inside of it. So we can go test our hypothesis. We're going to go put a little platinum down on top of the specimen. We're going to get the sides milled away. We're going to use the electron beam to kind of image that cross section, and we're going to do an iterative milling and imaging procedure until we generate a 3D structure. This is that same area where I've trenched out on either side of the bubble. We got a little bit of a platinum cap to kind of smooth everything out and protect it. And we're going to go through, and in this case, we collected 365 images going from basically the front of the area to the back. We used a software called MyPar, which we helped develop in a previous life to do the image processing. And so here's kind of an enhanced view of that area of interest. I've got a movie here, so we should be able to get the movie to play. Oh no, it's not playing. See if we can fast forward through here. So you can see as we take the successive slices through the bubble, we can make a movie of all the slices. We go all the way through the material. We can kind of see the different layers of the cuticle as they envelop the bubble material until it's all complete. Kind of tails off to the end. Now, as we move our reconstruction forward, we're going to go back and we're going to do image processing to isolate each one of the sections of the bubble material throughout the movie. This is a combination of binary thresholding, isolating the features, cleaning them up. Now we're going to go take that threshold data. We're going to put that back into the visualization tool to create a 3D model. And so now we can see how that bubble material is actually, what the shape of it is and what's happening in between those different layers of the cuticle. This gives us, the researchers that were working on this, a much better view and understanding of how this bubble is manifesting, and a little bit better view than they had where we're just looking at a simple cross section cut with a razor blade. So you can actually see a couple of different layers of the bubble forming in between these different cuticle layers. So again, we had found these bubbles, and just a little plug for there are things we can do in the FIB here. We can find these bubbles and isolate them. We can trench on either side of the material. Instead of doing a serial section, we can isolate this material, thin it down, extract a lamella, transfer this to the transmission electron microscope, and then perform EDS and electron energy loss spectroscopy to identify the chemical species and where they're located. This turned out really, really well. The paper published here, if you want to read all about it, in the X-ray CT and the other techniques that were applied to this problem. It's here. I can provide a link to the paper at the end if anybody wants it. So what else can we do in the FIB? We're really talking about serial sectioning today, and kind of excited about serial sectioning. I wanted to bring up something that I haven't done in quite a while, which is electron energy loss, or I'm sorry, this is the electron backscatter diffraction and serial sectioning. So this is a technique where we are going to mill the sample, and then the sample actually rotates around, and then we can collect an EBSD pattern, and that's going to give us the grain orientations of all of the features inside of a microstructure. And so a lot of times, say like titanium alloys, it can get a really complicated structure, and it's not intuitive looking at any 2D slice what is going on with all the different plates that are inside that microstructure and how they manifest themselves into this the 3D morphology. So here we're able to do the serial sectioning, where on each successive slice we don't just collect an image, we collect an entire EBSD map and put that data together. So this gives us the ability to look at these different plates in this Ti 6246 alloy, and it's not entirely clear how these plates interact with one another, but once we isolate them using the crystallographic information, we can now see this map and how the different features interact with one another. And they have a profound effect on the strength of the material, as things like dislocations move through the metal as it's deformed and loaded. Those dislocations can either easily or not easily transfer from one type of lath or one orientation to another. So as metallurgists and engineers and in processing these materials, we try and tune some of the alloy chemistry and also the mechanical processing steps to modify the structure to get the desirable processes, the desirable properties. But it's not entirely clear when you're just looking at the material, what's the rule set? Like, how do these materials go together? So using a 3D serial section can be a really, really valuable tool in understanding these rules and getting to know the material. So it's quite a bit different than looking at the bubble and the bubble hair, where, like I said, you can look at that bubble and you've got already kind of an idea in your head what you think that looks like. This is a case where we look at these laths and we really don't know, how they're interacting and how they produce the total structure. So this was a lot of fun to put together. So the limitations, if you looked at the 3D EBSD data set that we collected, you had about 48 hours of data collection time. But in removing that material, we only had about 4 hours of milling. So I think the entire run from the beginning setup through the total data collection ended up being somewhere around 53 hours. So it ran over an entire weekend. And this really made it clear that in this situation, the system is acquisition limited and not milling limited. So even if we were to have a higher current source or a plasma beam FIB, In this particular situation, it's really not going to speed up that process because we only had about 4 hours of milling in there anyway. The limitation was in the EBSD collection. So we don't have this set up on the FIB right at the moment. It was very clear that the generation of EBSD camera that we were running at the time is just not fast enough. It's practical to do this sort of experiment, but the speed is not there. So we're actively looking to increase the capabilities of the EBSD system for the dual beam FIB so we can be able to do more of these experiments in the future. And it's a little bit my hope in the talk and the discussion here to find any groups that are interested in doing this sort of analysis. We might be able to work together and do a little bit more of this work in the future. One of our current focuses is now to do serial sectioning and processing of the materials at cryogenic temperatures for doing soft materials and biomaterials. And so we have a quorum cryo stage, which allows us to plunge freeze and process cells or soft materials into a vitreous state, and then transfer that onto the stage where we can go mill it and image it. So if we look at the room temperature processing, where we had a small sample, metal sample that we had spent a lot of time cutting, grinding, polishing, and then attaching that to a pre-back tilt holder, which allows us to do the imaging and milling very readily. With a cryo sample, we don't really have the benefit of being able to rotate very easily. And we're not going to be able to go take that sample necessarily to a polishing wheel and polish it into the optimal geometry. So what we're going to do is we're going to process the sample in the microscope like we did with the hair sample. We're going to trench away material to expose the region of interest, and then make some relief cuts on the sides to get rid of redeposited material. We'll be able to do our imaging directly. This is where, if you have the access to a plasma source FIB and can remove a lot of material, this will allow you to get to that region of interest very quickly. However, once you transfer back over to the regime where you're doing the serial sectioning, you are going to probably end up running at roughly the same speed as if you were the gallium FIB. And there's the trick that when we're using lower beam currents for, like, say, the serial sectioning, trying to remove 10 nanometer or 30 nanometer slices, you want to use a lower beam current, and the smaller beam currents are actually that's where the source is sharper and brighter with the gallium FIB. So there's pluses and minuses to both systems. We'd like to have both at CMAS if we could, because they're very complementary tools. So you can see here on the 48-hour data collection, we ran roughly 100 nanometer slices. On our hair sample, we were running around 30 nanometer slices. And it was about a 6-hour data collection just for the imaging. And if we had wanted to collect, say, 10 nanometer slices, we would have used a slightly smaller beam current. So our milling time might have increased from maybe 30 minutes of actual milling time to maybe an hour or so. But the imaging acquisition would have been gone from maybe 6 hours to upwards of 18 hours. So in this case, we're still acquisition limited. It's more balanced than in kind of an exhaustive data collection like EBSD, but it's still not necessarily limited by necessarily the milling rate or the beam current. So that's mostly the points I wanted to talk about. We've got TEM and cryo EM talks coming up here in the future, and I want to kind of go back and see if anybody had any questions about the serial sectioning and the dual beam FIB.